my screen very quickly. Okay, so for those who just tuned in today and didn't join us yesterday, I'm going to do a very quick introduction. For those who are tuning in to us for the second time, you might have heard this already, but it's okay. Let's do this for the first timers. Okay, welcome to our Free Tree Society Live Talk Series. This is the second of the three-part talk series of Wild, Weird, and Wonderful with Dr. Chan Ziyang. Now, yesterday we explored quite a few cool things. Let me show you this cool slide that Dr. Chan actually made himself, which gives you a little snippet of what to expect in these talks as well. Now, yesterday we explored Beauty is the Beast, where we talked about some really cool birds and the way they survive and how they adapt, as well as some other flying creatures that we might have seen a lot but not really noticed, like butterflies and moths, and especially caterpillars, and the way that they have evolved in very strange and wonderful ways to protect themselves and to, I guess, to survive in what is becoming an increasingly dangerous world for insects to live in, especially when you consider how much human habitation has taken over the urban landscape. So we're not sharing our habitats too well. So these talks are also meant for you to understand the creatures and the plants that we share our spaces with a little bit better. And hopefully with this new appreciation, you can go about your days with a newfound love for nature and a newfound respect as well. So today, this is December 19th, Tuesday, and we're going to explore bizarre botany. So for those who missed out on the little promo that Dr. Chan and myself did on for this particular episode, here is a very cool video featuring a very cool plant. And you'll need to turn up your volume to listen to this, because it's not just about the sound, it's about the way the plant reacts to the sound. Okay, so take a listen to this. Just let it load. Internet, don't fail me now. Jingle bells, jingle all the way. Oh, what fun it is to ride in a one-horse open. Wow, that's really cool. Hey, jingle bells, jingle all the way. Oh, what fun it is to ride in a one-horse open sleigh.
Go.
Wow. I
Okay.
see it. If you guys don't see it, you need glasses. It's even
Okay.
responding to my voice as I'm speaking right now. That is
Yeah.
amazing.
Okay. Good.
Awesome. Okay, so that was the telegraph plant, and Dr. Chan will be talking about that in just a little bit, and why or how this plant reacts to sound stimuli. Whether we know a lot or not about it, we'll learn a little bit more later. But before we begin, as always, because we're working with the wonderful Dr. Chan, you know there's going to be some demos that you will be watching live, which are things you should not replicate at home. So here is a disclaimer for you to watch and read. Okay, so again, this is the same disclaimer we'll put throughout the talks. Please make sure whatever that you see today remains as knowledge. Okay, today especially, Dr. Chan will be working with some plants. There will be some some chemistry demonstrations that he is a scientist, can perform, and you as a non-scientist should not try. Okay, and if you do try to do it, remember it is at your own risk. It has implications to you that we will not be responsible for, such as injury, rashes, irritation, all these things. Okay, so he's very careful in the stuff that he does. And again, I remind you, he is a trained scientist. He knows what he's doing. We are regular people and we do not. Okay, so we do not take responsibility for what might happen should you replicate this at home. So let's just enjoy, watch, and learn. Okay, now with that out of the way, I'm going to hand it to you, Dr. Chan, to take over with bizarre botany.
You disabled me to share screen.
I disabled you to share screen. Oh my goodness. How did that happen? Okay, now you can share screen.
All right. Good. Hope you all can see. Let's go to the slide. Whoa, this is weird. Okay. So, good evening, everyone. And today we will be talking about plants. What can be so weird about plants? So, test yourself and see how much you do not know about plants. Let's start with this. Okay. So, I think many of you would have seen this in parks and gardens, right? This is a cool looking flower, and people like me now will call it the bed lily. But mind you, that is a misnomer. This is nothing of a lily. This has no relation with lily whatsoever. So this bed flower, as I'm going to call it, is actually related to the yam. You know, the ubi, the yam, the edibles and poisonous. Yeah, it's more
Yeah.
related to the Dioscorea yam. Okay, so the first glance of this flower is really weird. Okay, now let's look at some common species here. We have the white bed lily, yeah, and the green bed lily, and there are quite many other species as well. They are native to Peninsular Malaysia, Malaysia in general. Okay, and there are still some species like the cristata, which is quite complicated even to botanists. Okay, so regardless, let's look at the general characteristics of a flower. Okay, this is an inflorescence. We have the petal, and the flower, actually, in this case, we have the female part, stigma, style, and ovary, and also we have the male part, the filament and the anther, which contains the pollen. So this is our conventional, stereotypical flower. Now, how about the taka, the bed lily? Okay, so look at this. The bracts, okay, gets expanded to become wing. Okay, so this tiny bract here becomes expanded to become a wing. And the bracteals, which are these tiny, fragments, they become whiskers of the plant. And the actual flower of the bed lily is here. This one, can you see? 1, 2, 3, 4. The drooping one is the old flower, and the standing upright one is the new flower. This is really strange for a flower, yeah?
Yeah.
And let's look at the individual flower, which is this part here. Let's suppose this is a new flower. This is an open flower. We have 6 petals. Can you see? 3 large and 3 small. 1, 2, 3, 4, 5, 6. And at the center, we have the stamen and also the stigma. The male and female are compressed together here. Can you see this mushroom-shaped structure? Okay, and that is the female part, the ovary, the style here. And then this oppressed part here with this yellow dots, those are the pollens. The male part is here. And so you can see in this flower, it has this, ability to prevent self-pollinization. This is like so oppressed structure, am I right? So it's really weird about this flower. First, it has dull color. It's not really all that colorful. Okay, it has no nectar whatsoever, and it has no apparent smell. I mean, you can go and smell it, nothing. And it actually lacks visitation from bees and butterflies. Okay, the conventional pollinators, they're not interested. And the flowers actually tend to get eaten or damaged. It's not all that poisonous as well. So the question is, who is the pollinator? Who on earth pollinates a flower that is so weird? Okay, and it turns out that this is quite well studied. The pollinators appear to be female biting midges. Midges are tiny flies that bite you, and then the female, it's like a mosquito really, it bites you and it sucks blood. So it turns out that based on the studies in Singapore especially, can you see that? All these tiny, these tiny flies, they are midges, and they are mostly female. Okay, lots of them, flocking towards a new flower. Remember, the new flower stands upright. Okay, and you can see these midges, when they visit the flower, they can go inside the cramped space, and then they, get attached by pollens. Very strange, isn't it? So they are predominantly female. Okay, so that means this is really profound, yeah? So why does the flower attract biting insects? It's like using mosquito as your pollinator. And in some papers, they actually put forth a very daring, but I think tantalizing hypothesis that the bed lily, you see all those whiskers, right? It could be a form of floral mimicry. So the plant could be trying to mimic, like see this whiskers, it could be trying to mimic some animals with whiskers. Perhaps it also emits some kind of animal scent, and that is how it is able to attract these flies. And speaking of which, okay, go back here. Without biting midges, we won't have chocolate, true. So the cocoa flower is also pollinated. Yes, wild, weird, and wonderful, is also pollinated by biting midges. Yeah. You see, all the flower is trying to prevent self-pollination. So in this way, it is, evolved this intricate, weird relationship with fighting midges. Strange. And without them, you won't even have chocolate for Christmas. Okay, so very tantalizing hypothesis. Is it possible that the bed lily is a type of floral mimicry for warm-blooded animal with whiskers? Now, again, that's a hypothesis. We're doing science. We need to do experiments. So one way to experiment is that maybe we have to trim away the whiskers of the newly formed bed flower and then see whether or not they perform better or worse or neutral. Okay, so that's one way to do experiments with it. And this is evolution by sexual deception. There are well-known cases of plant mimicking an animal, and this is especially diverse in the orchid family. So here we have an ant orchid. These are not native to Malaysia, but just to show you how remarkable plants can be. So these two here on the left, they are orchid flowers, and you can see that this one is trying to mimic an ant, and this one is trying to mimic a bee. And they can even emit chemical signals of the insects so that, real insects get fooled by the flower and then they come in and then trick to carry pollens. Yeah. Very smart. And again, remember yesterday we talked about butterflies and how we have polymorphs, and this kind of evolution is happening in plants too. So we do have plants, this is a single species of plants here, okay, which can mimic a fly. But also we have the non-mimic. So we have this competition between the mimetic form and the non-mimetic form, and it really depends on the overall ecology to their success. Okay, so evolution is a dynamic process. Remember this. And so that goes on to very weird looking flowers. And this one on the left is, I think everybody knows that, okay? And this one on the right, it already looks very funny, but unfortunately, it's not flowering at the moment. So you have to watch the promotional video to look at it live. So sapromyophily, who needs bees when you have flies? So these are flowers which do not smell good, okay? Sapro means rotting, myo means fly, phily means love, okay? So these flowers attract weird scents, okay, to attract flies. Roughly there, I'm sure if you've been near one, it smells like a rotting corpse. And how about this one here? This is really gonna get very awkward, but believe me, now let me just do a simple PG-18, by the way, disclaimer. So kids, if you are watching, you may not fully understand what is to come, but that is okay. Don't worry, when you grow up, you'll appreciate the beauty of nature, of humans and plants alike, okay? So don't worry. Now, I'm sure you all have, gone through some forested parks or gardens, and all of a sudden you smell, oh my gosh, why does it smell like human male bodily fluid? Okay, so why? And this is so strange, isn't it? Now there are many plants that actually smell like this. You see, even in the it even comes out in news. Your street probably smells like desert right now. So it might not be, in spring, but here we may be able to smell it all year long. Okay, these are the species that smell like really funny. Cassinopsis, Stemona, and then this red palm in particular. Now this one is a very common in housing areas. So when it's flowering, you will smell funny things. And pitcher plant as well. So these plants, they can be counted as sapromyophily, okay? They attract flies, but then they have evolved this smell, which we humans coincidentally find very weird and awkward. So scientists love to study weird and wonderful things. So this is a legitimate, very well done paper from China, which has tried to solve these questions. Maybe some of us has been perplexed for quite a long time. Deciphering the chemical origin of the semen-like floral scent in tree plants, okay? And it is due to this single molecule called pyrrolene, okay? So don't worry, chemistry is back, but I'm gonna try to make it simple and fun. We're not made up of proteins, right? We eat meat, we take proteins, and we digest proteins into amino acids. So amino acid is this is one amino acid. This one is by the plants, by the way, we don't have this. And then these amino acids can be transformed. The amine group is going to be transformed here into something called 1-pyrroline, okay? So this is a simple molecule. See, 1, 2, 3, 4, 5, we're going to look at it. And this is the molecule which the scientists found that is responsible for the weird smell. And what is even weirder is that this one pyrroline, if we modify it just a little bit, the smell becomes pandan. Okay, I'm sure we all know the pandan smell. And then it's just a few atoms different. And if you really think about it, the two actually carry somewhat similar note. I hope I won't scar you, but that is it. So let's look at this molecule. I really want you to appreciate the wonders of phytochemistry. So let's go. There we go. So this I have here is the molecular model of the semen-like smell. It's a simple molecule. You see, it's a ring. So it's like a snake biting itself. 1, 2, 3, 4, 5. And the blue one is the nitrogen. Okay, just remember this shape. So this is the funky, funny smell. And this here is the pandan smell. Can you see the red one? The red one is oxygen. So it has an additional oxygen. So let me try now to overlap the two molecules together. So you can see this part overlaps well, am I right? So leaving only this part that is different. So this group here with oxygen, this group is what we call an acetyl group. So that is all the difference between the semen smell and pandan smell. Weird and wonderful. So I hope I settled some of the questions that you all may have when you smell funny plants next time. All right? So this is the beginning of our phytochemistry. And yep, if you want to learn more about phytochemistry, refer back to my talk last year. But now let's play another game, okay, of roulette. So let's suppose one day you are stranded in the wilderness of Malaysia, not actually wilderness, but suburb, or when you are, or maybe when there's something that happened, you have a choice. Okay, there is this pink berry and there is this blue berry. Which one would you eat if you are hungry? Which one do you think would you eat? Okay, 50-50. Take a guess, Rina.
I'd go for pink.
Okay, so let's see if the pink is edible. Okay. Sorry, Rina, the pink is the forbidden fruit. Okay.
Oh.
This pink one is actually Keratia mollissima. Okay, it's a beautiful one. And then this pink berry, it actually belongs to this plant, which is related to the grapes. Yes, the grapes are edible, but this version of a grape is related to the grape. It's the Vitaceae family. And this one is really a no-no. Why? Because in fact, I talked about it last time. So if you squeeze this fruit or eat this berry, this is what is in the berry. Crystals of calcium oxalate. So if you like, inside these berries, okay, we have crystals, okay, or kidney stones. Okay, so if you like this berry, grows kidney stones in itself. So when you eat it, all the kidney stone crystals are going to poke your mouth, are going to be causing severe inflammation, sore throat. And if your airway swells, your throat swells, it may even kill you. So I know people who have tried to eat this because it's so, so attractive, right? So some people try to taste it. Just a little taste can give them a whole week of sore throat.
Wow.
Very, very dangerous. It belongs to the same family as grape, but then this one really, take this toxic crystal to the extreme.
So it's similar to the Dieffenbachia, right? Because I remember this from your last talk as well.
Yes, it is similar to the dumb cane, right? But this one is way more serious, and we're gonna look at it now. Let's hope we can do some experiments. Today will be quite easy. Got this. So can you see the fruits here? It's very beautiful, right?
Yeah, they actually look really edible.
Yeah, they look like really nice, but then trust me, we're gonna look at how nasty they are now. So let me pick one here. Let me pick one. I'm not gonna eat it, okay, but I'm gonna put it in this mortar here. I'm gonna assume that we eat it, okay, by grinding it up, right? So I hope it won't spill. By the way, if you get this into your eye, it is the most unpleasant, most scary thing ever. Imagine all those crystals in your eye. So I should be wearing goggles for this, but
what if it's contact on skin, doctor?
It will be very, very irritating.
Okay.
Worse than the caterpillar, worse than the tarantula, and because this is microscopic crystals, it's gonna get stuck. Especially in the eye, it's bad. Okay, so you remember the Dieffenbachia, it's a dumb cane. So back in the colonial era, very nasty people use it to torture unruly slaves. Okay, bad. So now this is a microscope slide. We're just gonna put one drop, just one drop of this juice onto the slide. One drop. And let's see if we can put it under the microscope and see what is happening. So disclaimer, while I set up the microscope, please do not try to eat this plant. Please do not try to, have fun with it, in funny ways to torture people. I don't like to see that. So please, yeah. So let me try to set up the microscope and we see whether or not we can observe crystals.
Yes, guys, please remember, just take a moment to read this while Dr. Chan is
very common, yeah. So you may encounter it in gardens and parks. And so if it ever crossed your mind whether or not the plant is edible, no, don't even think about it.
Yeah, and don't even squeeze it in your hand since you're not even supposed to touch the juice of this berry. So as cute and juicy as it looks, don't eat it. You don't know.
Either choice, unfortunately. Okay, so we can see the crystals. Let's get it. So that's the. Can you all see it or no?
No, you need to switch your camera. Yeah.
Let me just put it here. Okay, so that's the microscope. And let's see if we can see crystals. No guarantee this is live, but let's go. Can you see that? The furry things.
Like tiny hairs?
Yeah, like tiny hairs. Those are actually crystals. All those are the kidney stone crystals, which are extremely nasty. Okay?
That's just from one drop.
Yeah,
One
that's
drop
just
can actually do a lot of damage,
so
right?
imagine if you eat a lot of it, you're gonna suffer. So imagine if you just eat just this, okay, suffer. Imagine if all those goes into your eye, it is very bad as well. So let's move on to another plant here. Okay, while I'm still here. How about the blueberries here? This. I think quite many of you would have seen this in parks and gardens as well. It looks like a lily and it has this blue fruits. I hope you can see the bluish berries, very nice. These blueberries. And how about this? This is called the flax lily, sometimes called the nodding blue lily or Dialina ensifolia in its Latin name. So is this edible? Now, there are people who have eaten this before, and indeed the blueberries are used to make a blue dye, much like a bunga telang. We're going to look at that as well. But is this plant safe? Okay, you may put your bet on this one, but this is a very curious plant, which, perplexed even me, okay? I have been, perplexed by this plant for a long time. It's only this year that I finally figure out whether or not this is a safe plant to eat. So let's go. Now, Dialina ensifolia. There we go. So this blue lily, okay, according to traditional Malay herbalists, is actually a nice, medicinal plant. Again, we're not here to, promote any plants or things, but according to traditional Malay ethnobotany, this plant, this lily here, is actually quite beneficial to humans in some ways that it is anti-inflammation, and even postpartum women can take it as a tonic. The berry is used to make a blue dye and it is safe. Okay, but according to traditional Chinese medicine, this plant is absolutely deadly. Okay, especially the roots. Okay, it is so deadly that it's used as a rodenticide, and it is said to be fatal to cattle and humans alike. Wow. So that is, can you see? There's a dilemma here. And even to scientists like myself, a young phytochemist, I detected not the usual kind of poisons. I actually tried to study this during my undergrad, and then I failed to detect any sort of usual toxins. So for a long time, I thought, well, maybe the Chinese got it wrong. Okay, because apparently here in Malaysia, this is a medicinal plant, but turns out the one that's wrong is me. But before that, let's look at blueberries, shall we? So since kids are here, you all know what's acid and alkali, right? And litmus paper. So let's say we have this blue litmus paper. What happens when it comes into contact with an acid? So this is nature, this berry here is nature's, what is that? Litmus paper, way to check pH. Let's see. Let's see. We have time today, don't worry. Right. So I'm gonna have, it's gonna pluck one blueberry here. See that? It's a blueberry. I'm just gonna squish it onto this paper here. Can you see blue color?
Yep.
Yep. So now this is gonna become a pH indicator. So if you learn in school, oh, see the seeds are dark black like diamonds, very nice. Okay. So anyways, this is the blue juices, and this is the same as bunga telang. So what happens if it comes into contact with an acid? I have some acid here. This is diluted stomach acid, if you like. So let's put some acid in here and see what happens. Can you see it becomes red?
Yeah.
Let's see. Can you see it becomes red?
Yeah.
So that is nature's pH indicator, anthocyanin. The same chemistry that there is in the bunga telang. And this anthocyanin, I've talked about it, it is safe to be consumed. So the berries are actually known people who have eaten these berries before. They are sour, but generally, I think the berries are safe. But when I looked into, really, really old literature, like those, conducted in 1985, they have actually successfully isolated a very, very deadly toxin from this plant. Yes. So this plant, this Dilena, this blue lily, is indeed poisonous, but conditionally so. And this plant is one that's really moody. It cannot decide whether or not it wants to be poisonous or non-poisonous. So at some time, the plant is poisonous. It makes a very deadly toxin. We're going to look at it. At other times, this plant is just benign. So there is really no ways for us to find out whether or not this is poisonous. The toxin is called stepandrol, and this is isolated by a group of scientists from Australia. And they have isolated it from a related plant, okay? This is related to the blue lily, the blueberry. And then this plant is really serious, okay? You see, it's called the blind grass. And if animals eat it, it's going to cause weakness in coordination, multiple organ failure, blindness, and death. Even if you survive, you can get blinded. Very, very poisonous. And the toxin, if you like, don't worry about it. The toxin, if you like, is like a recyclable item. So this plant has a toxin that is like paraquat. You all know paraquat? The very nasty herbicide. People drink it to suicide. Just one sip is lethal. Yeah, it is like paraquat. So this toxin, if you like, it can be recycled by your body. So every time your body recycles this toxin, it generates free radicals. It generates things that cause damage to all sorts of organs and genetic materials and proteins in your body. It's irreversible. Very, very poisonous. And based on this paper, okay, the dry root of this blueberry plant, if you like, and if we expose it to iodine vapor, it will give persistent gray colored spots. Okay, so this is one way to test for the presence of this toxin. And as I'm aware, now we're going to do this experiment. As I am aware, no one has ever done this experiment on Malaysian Dilena. So here we're doing it for the first time. So let's carry out an experiment that was conducted in 1985 and see whether or not our Malaysian blueberry plant is poisonous. So again, you wouldn't want to do this because it's dangerous. So before I begin, let me show you what I have done. Right here. Okay, so if you remember my last lecture about phytochemistry, we talked about this process called chromatography, and it's very expensive. So chromatography allows us, phytochemists to separate individual components out of plants. Now, I've carried out this experiment earlier today. So here is the chromatogram, okay, of this plant. So on this plate here, if you like, so on this plate here, you can see, so on this plate here, I have already experimented with, this is the root extract, by the way. So this is the root. I've extracted it with a solvent, and then I have run it on this TLC chromatogram. I have separated the toxins. So if there are toxins, the toxins will be stained here, like, you know, crime scene fingerprints. You know, you watch all those crime scene movies, right? So to detect fingerprints, they have this smoke that comes in and then they stain the fingerprint. So we're going to do the exact same thing, but just phytochemistry. So let's go. I have a flask here. Empty flask here. And this is iodine crystals. Iodine is an element. It's going to become very beautiful later because it's a purple vapor. So I'm just going to put some iodine crystals into this flask here. See? Right. That's more than enough. Overkill, really. And then I'm going to take a fresh chromatogram, TLC chromatogram, and put it into this flask. Now to make the iodine vaporize, turn into vapor, I just need to gently heat the crystals, and you'll see what happens. Okay, it's very pretty, this reaction. Not reaction really, this iodine is a solid. So when you heat it, the solid is going to sublimate. So iodine is going to change from solid state to gaseous state right away. Can you see purple vapor starting to form? Very beautiful, isn't it? Oh my gosh. Okay, that's overkill. Can you see purple vapor starting to form?
Yes.
So this is like, you know, CSI, we're staining the molecular fingerprint of this plant, and this is really an overkill. Goodness. Can you see that? It's very beautiful. So this is how very preliminarily, we scientists screen for certain phytochemical compounds in the laboratory. Okay, so can you see the plate has changed color? And can you see some what is that? Some spots starting to form. Can you see that? Right? So according to the paper, if the toxin is present in this plant, we will observe a persistent gray spot later. So now if you excuse me, I'm going to let out the purple vapor. It's quite toxic. Again, don't do this at home. And we'll see whether or not there is toxin. I don't know the result really. So this is the first time I think it's been done in Malaysia. Okay, so let me just get rid of the vapor.
Okay. I hope none of you at home have iodine crystals just lying about at home. And if you do, please don't do this at home. Do it somewhere as well ventilated, preferably in a lab with some safety materials or equipment as well. I think Dr. Chan went outside to release the vapor. Oh, he's back.
Okay, so this is the newly stained fingerprint chromatogram of the plant. So we'll leave it to slowly come off. The yellow color, the counters, they will come off. And then later we'll see if there are gray spots, persistent gray spots here. If there are, it means that very likely, now this is preliminarily, but very likely that our Malaysian blue, nodding blue lily, flax lily is potentially very poisonous. Okay, so this takes some time to come off, so we'll come back to it later. Let's continue.
Okay, but that's just the root, right? So the berries are not.
Berries, as far as I know, should be okay.
Okay, so it's just the root.
This looks so tasty, right? So inconsistent toxicity, highly poisonous like paraquat, no antidote available. If you eat its root, I don't know what will happen to you, probably very bad. And for lay people, there's probably no reliable methods, okay, to tell whether or not this plant is actually poisonous or not. So to err on the safe side is not worth eating, especially the foliage and the root, okay? So that's it about this plant. So the question is, the original question is, if you are to eat any plants, the wisest answer is don't eat any if you are not sure, okay? So never mind, since this is a leisure talk, we're going to talk about survival. So let's say if you really got stranded in the jungle and you have nothing to eat, hungry, and you come across some berries and plants that you think, oh my gosh, I need to eat something, right? So here are some tips, okay, that I think you can do. So if you are stranded and in this survival situation, look for common enough plants. You're going to have to find plants or berries which are common enough that you can consistently find. There's no point finding plants which are rare and you don't have it to eat anyways. So no poisonous traits. I've talked about this in the last lecture. Any plant with milky sap or spiky seed pods, all these usually mean poisonous families. No mushrooms, okay? So mushrooms, even the experts got killed by it. No mushroom in survival. Don't even eat any. And the way you can do it is like this one. So let's see if I want to test this pink berry is edible. I can take some sap, take some juice, and rub it on my skin, okay? And see whether or not there are irritation or unpleasant reaction. So if there are any, you will not want to eat it, okay? You can just discard it and try another plant. And then if you tested it on your skin and on any surface, you don't feel anything, it looks benevolent, then maybe you can test it, but not swallow. You don't swallow it, okay? Just taste it. So if you have any burning sensation or just weird sensations, then just spit it out and try another plant. Okay, so if you pass the level number 5, you can indeed chew the plant. Okay, this is testing. Yeah, this is how scientists survive in the wild. So you can chew the plant for a few minutes, then spit the juice. Because when you chew the plant, your saliva is going to extract some toxins out and you can spit out the toxin, if any, okay? Then you swallow your masticate and observe for at least 8 hours that you have purging or any bad reaction. So if none, you can try larger quantity and repeat, and you have found yourself an edible plant. And indeed, I think our early ancestors, herbalists, okay, pharmacists, early pharmacists, they have done this on themselves, okay, to find out, which plant is edible and what constitutes medicinal effect. We owe this type of sacrificial spirit to our ancestors and herbalists. Pharmacy, drug, the word drug comes from plants. Drug literally means dried up plants, okay? So this is something for you to keep in mind. And yeah, back to this. Now, the only reason I cannot do this live is because this dancing plant, the telegraph plant, sleeps during the night. So if I bring it here, you'll just see it drooping anyway. So it's very nice that Rina has indeed come out with this very nice promo. I love it. It's so, comedic. And this plant, believe it or not, the telegraph plant has inspired prominent scientists, zoologists, botanists all over, including Charles Darwin, who wrote about it. See, in his book, The Power of Movement in Plants, many years ago. And back then, we don't have, smartphones and macro photography. He has to painstakingly draw the plants and explain how it moved. Can you see it? It's here. And here I also have a, time-lapse version of this plant I can show you. It's really, really weird and remarkable. Oh, I hope it's here. If it's not here, then too bad.
Oh, there it is, dancing plant.
Dancing plant. Okay, I hope it is what it is. Okay, let's see. Oh, what happened to my eyes? All right, so this is a time-lapse video. This is very intense, so you can see it moving. So even the big leaflet can move, but what is most remarkable is these tiny leaflets here. Okay, they move in this telegraph fashion, and that is why it's also called the telegraph plant. If you're old enough, you know what is a telegraph. If you're a kid, sorry, too bad.
Just
now,
Google it, guys.
Let's Google it. Yeah, technology. You see how old I am now. Okay, now this is how it works. So in the very tiny leaflets, we have this structure called the pulvinus. So in the plant leaflet here, this is the leaflet. You can see this rather swollen part. Look at my cursor, yeah? So this part is the pulvinus. And in the pulvinus, we have 2 types of cells, but the most important one is the extensor cells. So this cell is like a water reservoir, So it can either keep or let water flow out. Okay, it can keep a lot of water and it can also expel a lot of water. So the plant itself generates a kind of an electrical signal by moving ions in and out. Okay, like salt. The plant moves salt in and out, and that will cause the plant to accumulate or not accumulate water. So when the extensor cell accumulates a lot of water, it swells up and it causes it to extend. So if you like, it's just like men, if you get what I mean, okay? So Yeah, so there is a rhythm that's happening, and so the cell, what is that? The cell, how am I gonna put it? Okay, this is hard to put because it's nervous system. I can't claim that plants have a nervous system. So there is this rhythmic cycle of turgid and flaccid state. Okay, and that is going to cause, and that is responsible for the movement cycle of this plant. So that brings us to this question, which is why I was so I don't know how to put it. Does it then mean that the plant has a very rudimentary type of a nervous system, much like how our heart beats, right? There is a cycle, there is movement of ions. So this is a nervous. Does that mean that plants or certain plants have some type of nervous system? Now, this is highly debatable, really. I don't think so, but it does have some kind of a nervous type response, okay? And even when and in this article, the Venus flytrap and the dancing plant, if we apply human general anesthetic drugs like the inhaler, like chloroform, ceflurane, this kind of drug to the plant, and they become unable to move. Same way like humans. Yes, this is well known. You can go look it up on YouTube. If you apply anesthetic drugs to moving plants, they stop moving, right? This is well known. What is the mechanism is still being widely researched. But here, let me just put, some justice to plants, really. And this is a very groundbreaking study. Plants can even emit sound under stress, and then the sound can generate airborne information to other plants. It's just that we can't hear it. So this is what the scientists did, really. They stopped watering their plants, and they cut their plant, trim their plant, injure their plant, and then they use this ultra-fine microscope to listen to their plants. So the plants literally scream for help. It's just that we humans can't hear it. So follow this article. They have actually turned the sound to human audible version, and you can hear plants scream. So the scientists actually propose that when the plants are stressed, okay, or injured, tiny bubbles inside the plant's vascular tissue will explode or crepitate. And it is this tiny explosion of bubbles inside the plant that generates this ultrasonic sound, which can be heard by the way, which means recepted by other plants, and they start upregulating their plant defense mechanism. So this study to me is like, I'm sorry to vegetarians and vegans, when you eat your plants suffer badly. And it's just that you can't hear it. So I am say vegetarian and vegan, we do have a positive impact on carbon footprint, but as far as humane, I don't think so. Plants suffer a lot by vegetarians, okay? Go look up this study, it's real, okay? It's just that we can't hear it. Okay, so back to the dancing plant. 100 years, more than 100 years since Charles Darwin make it, superstar. No one knows why it moves like that. Okay, so there are many tantalizing hypotheses, but they need to be tested. So one hypothesis is that by moving in that circular fashion, it is a form of light tracking. So the dancing plant can track light intensity and move towards a position with better lighting. Is that the case? I don't know. In fact, it's native to Southeast Asia or potentially Malaysia. Okay, you can even buy its seed on Shopee. I don't know if it'll grow well, but if you would like to have fun with this plant, you can buy it anywhere. It's cheap. And then it could also be an anti-herbivorous behavior. So imagine if this plant is moving like this, predators or not predators, herbivores, insects, they don't like to land on moving plants. So could it be a form of anti-herbivorous behavior? Or perhaps this one is really tantalizing. By moving the tiny leaves, the plant can actually attract the attention of birds or mantis or jumping spiders to come and visit the plant. Okay, they feel curious, potentially they are prey, and this in turn, okay, deters insect herbivores. Could it be the case? We have no idea. And this is really an ongoing mystery, and I like it that way. Okay, so that's it about the dancing plant. This is short and easy. And here we're going to move to the weirdest of the weirdest plant here in Malaysia, okay, or in our urban jungle. The pitcher plant. This is the second part of my talk. Today will be light and easy, but let's look at the pitcher plant, because a lot of us now love to plant this, I hope, and then it's really strange. Have you ever wondered, like, why would plant evolve to become insect eaters, right? Why would carnivory evolve in plants? So if this pitcher plant or Venus flytrap is so successful, why not more plants turn carnivorous? Okay, so we're going to try to answer this question. And a lot of people will ask me, is there acid inside the pitcher's plant? How does it kill its prey? Okay, how does it digest its prey? Okay, we're going to look at that. So pitcher plant, its Latin name is called Nepenthes, and it means not sorrowful. So this is Carl Linnaeus, and he first described Nepenthes, and he was so thrilled, by the discovery of this weird, pitcher plant, and he called it not sorrowful. After this, Nepenthes is actually a medicine. They drink it and they forget about all the pain in the world. Okay, it turns out to be morphine, but no, this is it. And here we call it the monkey's cup. So now we're going to also try to answer, do you think monkey really drink from the monkey cup? And are there, and can you drink from it? And certainly there are monkeys out there. Here, the reason why I put up this slide is there are monkeys out there who goes and collect all this wild pitcher plant only to put rice in there to steam lemang. This is very bad, yeah? So anyone watching, if you are sure that this is non-cultivated, this is I think this is ampullaria, right? So please don't support this. This is bad. Okay, so there are monkeys here who like to do this kind of thing. So it's not good unless you cultivate it and then do this kind of unique food. Otherwise, please don't collect wild pitcher plants to do this kind of thing, right?
Agreed. Yes.
So the carnivorous pitcher, not all pitcher plants are carnivorous, but since we're doing the urban jungle, we're going to talk about the carnivorous plants, Gracilis, Mirabilis, and Alba marginata. Okay, so these three, they are deadly to insects. Okay, they have this fluid in their monkey's cup, fluid that can trap and kill insects. And we're going to look at what is it and how does it kill the insect. And is it an acid? So here I actually took pH paper, okay, and tested this is roughly about one pitcher plant, many pitcher plants from my friend's Gideon's nursery, right? And this is an extreme example. People always ask me, is it an acid inside? And in this case, yes, it is acid. And so much so, it is pH 1, okay? So we all know the pH scale. pH 1, and the pH scale, by the way, is pH scale is non-linear. So pH 0 or pH 1 and pH 7, it's 1 million times more acidic. So in this pitcher plant, it is 1 million times, more acidic than plain water. Okay, but most of the pitcher plant I have tested, most of them, okay, I've tested is around pH 4 to 5. And this one is really an extreme example. Now, since this is my Christmas talk, we're going to look at extreme example anyways. So what is an acid? Just now we look at an acid, right? So acid basically, in the most layman way, because last time is too much chemistry, this time there will be less chemistry. So acid, if you like, water is what? H2O, am I right? You give one more H to H2O, H3O is an acid. So for all intents and purposes, remember that H3O with a positive charge is acid. And pH is basically a measure of how much H3O is there in a given solution. So let's suppose we have 1 liter of stomach acid or 1 liter of this pitcher plant fluid that is at pH 0. How many H3O molecules do you think that there are? How many? Think about it first. 6.02 times 10 to the power of 23. Astronomical number, right? So that's very concentrated. That's a strong acid. And what acid can do is something called hydrolysis. Okay, so remember we are made up of proteins and proteins are made up of amino acid. Okay, so amino acid here made up protein. So if we subject protein to amino acid, the reverse is going to happen. The acid is going to break down proteins or this peptide back into amino acids. So this is a form of digestion. That is why the pitcher plant, the monkey's carp, and humans and snakes, many animals with digestive systems rely on an acidic medium to do digestion. Okay, those that eat protein especially, we rely on mostly on acid. Okay, and that is also why acid is dangerous. Okay, concentrated acid, it can do this to you as well, to your skin. And so let's look at it. Let's look at an example of acid and think about its implication in the monkey's cup. Now, this is definitely a dangerous experiment. Please do not do it. See that death if you do it wrongly or if you try and fool around with concentrated acid. Okay, it's not forgiving. So a lot of people have asked me, how can digestion of an insect happen? So here I have a video which I have. This experiment is so dangerous. I have to record a video in the film hood and then only to show you the toned down version of it. Do I have the acid digestion of insect? So this is what is going to happen in the monkey's cup. Okay, so there's a dead insect, by the way, it's not alive. So here's a dead insect, which I have subjected to a strong acid to stimulate what is happening inside the monkey's cup. Only it's sped up a lot. Okay, so this is a dead hornet, and this is a powerful acid. Okay, so this is digestion by acid. So you can see it's starting to bubble up. Can you see the insect exoskeleton and proteins that are starting to break down? Okay, so if you eat an insect, that is going to happen as well. So in your stomach, you have this peristaltic action, okay, which is going to speed up even more. Can you see that?
Yeah.
Yeah. So this is digestion. This is what is happening inside of monkey's carp, inside of you after your dinner, inside of especially snakes, right? We'll talk about that tomorrow. Snakes have especially powerful stomach acid. So this is part of what is happening in the pitcher plant. Okay, so after you see the insect is dissolved or digested, the nutrients, okay, it's going to go into solution and absorbed by the plant or absorbed by your body. Okay, this is how we work. Terrifying, isn't it? So let's look at pH 1 and whether or not it is, well, drinkable. Okay, so this is just to, this is just chemistry, but please, do not try this at home. It's dangerous, and as you'll see, I will have to wear some personal protection as well. So when I wear gloves, it almost always means something very dangerous is going to happen.
Yes.
Oh, look at that. Okay, okay. When your PPE fails you, that's one of the most scary things that's going to happen. So let me just put it on more gently. I don't want anything to do with this one. So here I have, a strip of tissue paper or cellulose paper. Behind it's just a Tupperware. Okay, I'm gonna just drop some acid here, just to show you what acid can do really, and so that you can appreciate the chemistry behind such substances. And I'm gonna have to wear this. If strong acid gets into your eye or your face or anywhere on you, very bad things are going to happen. So this is the same acid which I use to digest the bug, right? So I have some of it. Let's get ready. Let's see. So let's go. Can you see that?
Yeah.
It's starting to eat through. Horrible, isn't it?
Yeah, you can see it literally.
Yeah. It's terrifying, isn't it? Look at that. Look at it. Okay. I'm not going to tell you what acid this is. I don't want people to, I don't know, replicate this and injure themselves. But this is one acid that, you use it in daily life. But one of the most important acid known to humans. It's also a natural product, by the way. Volcano and acid rain has this, but in its concentrated form, it is extremely dangerous. So this one, if I'm to, test its pH, is going to be below 0. But here, I'm going to try to stimulate a pitcher plant, okay, by having some water. This is just water. And I'm going to drop this very dangerous acid, just one drop into this water, right? So it will probably give us, here's the pH paper, it will probably give us a pH of about 1, okay? So this is what we do use in the lab to check pH. So you can see here about pH.
Wow, it's already red.
Sorry?
It's red.
Okay, so it's pH of 1. Now, what will happen if, let's say, this is equivalent to the very, very acidic monkey cup, yeah? It can go as acidic as this. So what will happen, say, if I put my hand into this? What do you think will happen? All right. So let's do it, by the way. Don't do it at home. I'm just gonna I'm showing this to you just for educational purposes. So I'm just gonna put my hand into this. Right. Nothing much will happen. Okay, my finger will not dissolve. It will probably dissolve if I leave it there for 1 week. But then if it's a brief contact, it is okay. There's a reason why I am doing this. Okay. Now, this is just knowledge for you. So if you come into contact with very, very corrosive or highly, strong acid, you can always neutralize it with a weak base like this baking soda, yeah? So if you have cleaning products with strong acid and you got some onto you, you feel funny. So all you need to do is just to wash your hands with a solution of baking soda. Okay, that will neutralize the acid. Okay, so this is just some safety information for you. And while I go and rinse my finger off, we'll continue with the pitcher plant. Right. Now, as we have seen, even if it's pH 1, it is not all that dangerous. So the acid, okay, is not the main killer in the pitcher fluid. It may be very acidic, but it is not what is killing the insect. Okay, and indeed, acid alone, even at pH 1, takes too long to digest, the insect. And the Nepenthes need help from something called an enzyme. Okay, now let me go and rinse out my finger and then we'll continue. Just take some break. If you want to ask questions, then just put it down in the comments.
Okay. So he's gonna rinse off his hands really, really quickly. And in the meantime, I think back to the issue, and I see on the comments here, Tanji saying yes, lemang periuk kera is not sustainable. It's true. I personally have never heard of people who are cultivating it specifically for the lemangs. All that I've seen has been taken from the wild. So let's not support it, especially with the new year coming. Hari Raya is also coming in a few months. Please don't support this unsustainable business.
Yes. There's a reason why I put it. Okay, so the Nepenthes has acid in its arsenal, but the acid is not the main killer, and it needs something called an enzyme. Okay, do you know what's an enzyme? So an enzyme is basically any of a given protein, is a type of protein that's gonna help to help a chemical reaction to happen way faster. Okay, so you just look at this cartoon. So if the frogs represent a chemical reaction, any chemical reactions are going to have some barriers to them. So an enzyme reduces the barrier so that it facilitates the chemical reaction from happening. And enzymes, they are not used up or consumed in a chemical reaction. So to really show you, okay, what is an enzyme or what is a catalyst? You all know what is a catalyst, right? So an enzyme is a biological catalyst. Now to show you, wow, more chemistry demo to come. To show
Oh,
you
no,
what
that's fire.
Yeah, no fire this time. I hope there is none. So please, no fire. But we'll. I'm just going to show you what is the catalyst. Okay, so let's go. Lots of chemistry, Christmas lecture, anyways. So here, I don't know if you can see my whiteboard. Okay, so behind here, I've already written down this equation, but don't you worry for now. And here in this class, something fell.
I hope it's not acid.
The acid, thank goodness. So here I have a solution of hydrogen peroxide. I don't know if you all know what is hydrogen peroxide. Water is H2O, hydrogen peroxide is H2O2, right? So this hydrogen peroxide is unstable. Hydrogen peroxide is unstable. I'm just gonna, put a little, I think that is more than enough, goodness. So this hydrogen peroxide is unstable. It will degrade into water and oxygen gas. And even in this room temperature, it is slowly degrading into water, H2O, and oxygen, O2, right? So I'm gonna add a catalyst, okay, in here to speed up this reaction. Now remember, the catalyst is not gonna be used up, but it's gonna speed up the decomposition of hydrogen peroxide into oxygen and water. So imagine how fast that reaction can be. So here is without catalyst, and now I'm gonna add in the catalyst, which is here. This is another dangerous one. In fact, concentrated hydrogen peroxide is used as a rocket fuel and also in torpedo head. You definitely do not want to fool with the concentrated stuff. This is not even concentrated, but it's enough for me to show. So this is the catalyst. I'm not going to tell you what it is. I don't want people to go and injure themselves. That is enough. This is the catalyst. You do not need a lot of catalyst. How much reactant? How much catalyst? You do not need a lot of catalyst to catalyze a reaction. Very little catalyst because they are not consumed in the chemical reaction itself. Certainly want to be careful with this one. Oh no, I have to wear my gloves. Wait a
Safety
second.
first.
Yes. So when you see me wearing things like this, things are gonna get fun and also very serious, right? So just make sure that things don't fall onto myself and get my computer and electronics out of the way. Goodness. All right. Can you all see the flask?
Yep.
See the flask?
Yes.
I'm gonna add in the catalyst and we'll see what happens. 3, 2, 1.
Whoa.
Christmas, right? So that is how fast the reaction becomes. Look at that. So this is water vapor and oxygen gas. Yeah, Christmas class for you. It's still happening and it's very warm.
But the gas that's coming out of it, or the vapors,
Water vapor and you can just
is it dangerous?
vapor and oxygen. So I'm fine. I can just inhale all this and I'll still be all right.
Okay.
That's the reason why I do this. So now you have seen that very little catalyst can do a lot of work. So in the Nepenthes, in the pitcher plant, it has a lot of these enzymes that catalyzes the digestion of its prey. And let's look at it now. Dangerous, yeah. So if you do this experiment on an enclosed space, do you see how much gas is produced? In an enclosed place, it can just go boom. So don't do this. And so this is the catalyst, the biological catalyst of the pitcher plant. It's called nepentecene. So anything that ends with syn, pepsin, synsin, or ase, they are enzymes. And this is the type of enzyme called the aspartate enzyme. So can you see this ASP here? So this is a big protein, but the really important part is this ASP, aspartic acid. And it's red here. So if you like, okay, so this aspartic acid is using water. Can you see H2O here? It's using water to break down amino acid. Can you see this chunky part here? It's being broken down into 2 parts. So this is the digestion process. And can you see that the red parts here, they remain unchanged, right? So this is how the enzyme work. It facilitates hydrolysis under acidic environment. Okay, so the monkey cup has a lot of these enzymes to help break down different things. Now, carnivorous pitcher plants, they can break down insect exoskeleton. They have this chitinase and stuff. And there are also pitcher plants that eat dead leaves, and those can break down cellulose and plant tissues. Okay, it's a very diverse evolutionary success of this group of plants. And the pitcher plant also contains toxins. Yeah, so these are some of the potentially toxic substances called secondary metabolites. We have talked about them in my last year's talk. And the pitcher plant, sometimes they contain this compound called plumbagine and naphthoquinone. So again, this toxin, remember just now I talked about a toxin that can recycle itself, right? So the Nepenthes actually has very low level of same kind of a toxin, if you like, called plumbagine. And this toxin can help the Nepenthes to keep itself clean. Bacteria won't grow out of control. And that is why some people, they claim that if you have some injuries and you put the Nepenthes juice there, it can have some effect. Now, this is not proven, but if it indeed has some effect, it could be due to this secondary metabolite called naphthoquinone. Okay, it's just this double ring here, this molecule here, right? And this is, just explain how it works. No worries. And inside the pitcher plant too, we have symbiotic bacteria. Okay, so imagine if you're a pitcher plant, okay, you can't move, but you have this digestive system, enzymes, acid, and it's trapped. Whether or not bacteria is good for you in a pitcher plant. Now, it's a complicated case, but we do find that there are many bacteria in the pitcher plant, but also at the same time, some pathogenic bacteria are indeed inhibited by the pitcher plant. So here you can see, challenging the pitcher fluid with bacteria or yeast cause bactericide as well as fungicidal properties in the fluid. It makes sense, you see. If you're a pitcher plant, if your contents grows fungus or yeast or bacteria, that's bad for you. So the pitcher plant will have to find out some ways to limit, but not eradicate bacteria or fungus from its pitcher fluid. So microbiome. Okay, so here is how the pitcher plant or carnivorous pitcher plant really goes scary. Okay, there are some pitcher plant like the Rafflesia, which if you look at the pitcher fluid, it is very sticky. It's almost like honey. We can pour like that. Okay, so that is something called viscoelastic fluid. This is very well studied. Okay, and it's very, very interesting. It is like, playing Tai Chi with your prey. Okay, so if the insect goes into this viscoelastic fluid, if it moves a lot, it is like hitting a wall. But if the insect does not move, then it gets wrapped and dissolved or covered up by the fluid. It's really like playing Tai Chi with your prey. It's like honey, okay, and also rubber band, right? This is well studied. Okay, viscoelastic deadly fluid. And it is this viscoelasticity that helps the monkey cup plant to capture quite a lot of flying insects. So when the insects land into the liquid, they get trapped. But again, is this the killing force of the Nepenthes? Maybe not. The real killer in the killer fluid of the Nepenthes is actually due to what we call wetting effects. Have you seen this water strider before? Okay, these insects that can walk on water, and even if you put ants or flies or tiny insects on water, they won't sink, right? They will just float on the water, or something can even walk on water. And that is due to something called the surface tension. Okay, water, if you like, water molecules are going to pull at each other, and that is going to cause this interface, okay, to shrink, but also there's a surface tension. So that if you're very lightweight, you can make use of this surface tension. So in order to, prevent insects from escaping, okay, the pitcher plant also has a soap-like effect, okay, what we call surfactant effect, that reduces the surface tension of its capture fluid, the picture fluid. And this is going to really cause the insect to sink. It is like this. Imagine insects have all this on their exoskeleton, they have wax, right? They have this wax. So the pitcher plant can really wash away, if you like, it has a detergent-like effect that washes away the wax on the insect's exoskeleton. And that will cause the insect to sink real fast. And it is this sinking and drowning effect that really kill the insect. I've done experiments on it really. The pitcher plants, especially gracilis and mirabilis, can kill an insect within 20 seconds. Okay, if you want to do this experiment, you can do it. Try to drown an ant in water and see how long it takes. Okay, you may take forever. Okay, your ant may escape. But if you try to drown an ant in pitcher plant fluid, the ant may perish within 20 seconds. And that is mainly due to, just now as I explained, the viscoelastic effect, as well as, most importantly, this wetting effect. Okay, it's a soap-like effect. So it's same as you try to drown your insect in soap. Okay, they die really quickly. It's very efficient. So I hope I've settled how the Nepenthes actually kills its prey. And this is the study that how scientists do to find out. Okay, you may think that it's cruel, but this is how we figure it out. Okay, so you can see they try to sink an ant in water. This is just water. This is just water here. See, the ant floats, and this PF is pitcher fluid, and you can see the ant really gets washed up, and then it drowns. And this is the stickiness. Can you see the stickiness? The viscoelasticity is here. Okay, and this is most scary. So if they take the very, close-up microscopy of the animal, the insect, when they come into contact with the pitcher fluid, can you see all the hairs and all the exoskeleton are stuck together by the fluid? This is pitcher fluid. It's like, very dirty, muddy road, right? Scary. So this is how it kills its prey. Okay, so there is supposed to be a demo on this surfactant effect, but I actually killed my own pitcher plant, so I don't have pitcher plant to show you now. I hope in the future, because I have a friend, actually his name is Gideon, if you're watching, I will invite you again in the future. So in the future, if we are doing the pitcher plant again, I'll do this demo, okay? Alternatively, if I can do it, if I can get hold of some really good pitcher plant, then I'll do the demo and put it down in the comments. So here's a really special one, which I have it with me. This one here. Can you see a white line? This is called Nepenthes albo marginata. Okay, albo means white, marginata means margin. So this Nepenthes is quite common here, especially in Penang. Okay, you can see this. Can you see a white patch here? A whitish patch here. Why do you think this Nepenthes has evolved this special white patch? Okay, which is unlike any other. Okay, this is something that's quite common in the I think in around secondary forests or parks and gardens even. So this one, why do you think it has this weird pattern here? Okay, it has puzzled scientists for quite some time, and even me and even a lot of people, and they found that this white patch is actually a termite attractant. Okay, yeah, this has been published in Nature. So this pitcher plant has this very attractive to termites white patch. Okay, so at night, certain species of termites, they will find this irresistible, and they will come here and just gorge on this white patch, and almost all of them will fall into the deadly trap. So this pitcher plant, instead of slowly trapping insects over its lifetime, so in certain areas in Borneo, I think so, okay, it will just capture all the food it needs in a single night. Okay, this is some weird strategy, but then here in Penang, at least in Penang, from my observation, this doesn't happen. Okay, so how does this adaptation really work in habitat where termites or this particular species of termites are not so abundant? I don't know. So this is still a mystery to me, and I try to figure it out hopefully in the future. Okay, so this is really, if you observe this, try to look at the prey and try to look at what's inside of it. And speaking of what's inside of it, I've just shown you how deadly the Nepenthes fluid is. Am I right? And tomorrow, we're going to look at one creature. I'm not going to tell you what it is. One creature that can survive and even deep dive into this deadly Nepenthes fluid. Okay, we'll find it out tomorrow, nearing the end. So here's a question for you to think about. Only about 0.2% of all flowering plants are carnivorous. So why not, since this carnivorous plants look so cool, right? So powerful, right? So why not more carnivorous plants in the world? And remember, I said that in evolution, everything has pros and cons. So carnivory actually comes at a very high cost to carnivorous plants simply because they have to sacrifice their photosynthetic capacity. Look at the pitcher plant, okay? The pitcher plant is hardly, photosynthetic compared to, plant that can really have a lot of surface area in photosynthesis. Okay, there are many other factors as well. And this carnivory only favors plants that live in very, very, very nutrient limited soil. Okay, so it's not all that. It's like choosing, the less of two evil, really. So we're going to look into that soon enough. And if you have experience keeping pitcher plants, unlike me, I just killed mine because of experiments. So I'm going to show you why. If you this pitcher plant, you can easily over fertilize it. I have this pitcher plant given to me by one of my Dr. Tanji, if you are watching, and I have this pitcher plant. And then I watered it with I have a fish pond as well. So I accidentally watered the pitcher plant with my fish pond water, and that is considered over fertilization. And what happens after is that if you over fertilize your pitcher plant, it is not going to produce pitcher anymore. It's just gonna grow this very ugly long leaf, which is photosynthetic. So you can see the cost of carnivory is so high that when the plant has enough nutrient, it does not want to be carnivorous at all. And if you continue to, over-fertilize your pitcher plant, it will die. And that's how I killed mine, actually. I'm experimenting it with different fertilizers and see which one is most terrible to the Nepenthes. And indeed, it is the nitrogen. Okay, if you over fertilize it with nitrogen, that's very, very bad. Okay, so if you have never kept a pitcher plant before, I certainly encourage you to keep one, because you will see a tiny sort of like ecosystem happening. So when you can even see jumping spiders or geckos go near your pitcher, and then you can see you can try to study what insects it tries to capture and all. So if you have never kept a monkey's cup before, I certainly recommend you to keep one and observe the various evolutionary adaptations that this plant has, okay, to survive this poor soil environment. And here is the very interesting paper, which tells us, this is by Harvard, by the way, which basically tells us that, okay, the evolution of carnivory is more like the Hobson's choice, choosing the lesser of two evils in a bad situation, okay? So it is remarkable that carnivorous plants are here at all. So especially here in Malaysia, we are the center of biodiversity for this monkey cubs. Okay, there are many, many other species, and they do diversify. Not all of them, eat insects. Some of them eat poo as well. But those are really, montane or jungle species, which we may go into next time. So again, this is a marvel of evolution. The monkey's cup is definitely one of the weirdest plants out there. I've just, talked about a very teeny tiny part of them. So maybe in the future, we'll come back to this as well. So to answer the very, very, rude question, can you drink the monkey's cup? Okay, so unopened and fresh pitcher, it could be safe to drink. And if it is pH 1, then definitely it will be more sour than drinking vinegar. But then my friend told me sometimes it tastes bitter. I have no idea why. It could be due to the secondary metabolites. And I would not recommend you to eat the pitcher plant per se, because remember the toxin, the recycled toxin, imagine, not so recommended. And aged pitcher, okay, pitcher that is aged like the one I just shown you, there are lots of bacteria and decaying matter, and then we don't know what those bacteria or microbes are. It is not a very good idea to eat it, okay? So if you have gastritis, okay, or immunocompromised conditions, allergies, then I will certainly advise you not to drink or eat Nepenthes altogether. But there are at least some people or two people I know of that that drinks Nepenthes from time to time. Okay, so you can meet one of them here in the Penang Hill Monkey Cup garden, which has actually quite many of my plants here. I took picture from him. Eric, if you like, if you're watching this, thank you for sponsoring this part. And if you want to meet the person who has drank Nepenthes for I don't know how many years, then go visit him at the Monkey's Cup Penang, Monkey's Cup Garden or Kopi Hutan now, Penang Hill. Okay, you'll see a lot of nice species. And that's it for this time, the bizarre botany.
Oh, no, doctor. We haven't finished yet because Tanji, who's on the chat, and myself as well, and I think everybody else, we still remember that there's a chromatograph
Oh
that
yeah, okay.
and we need to see if it's turned gray or gray spots.
Oh yeah. Thanks for reminding. Okay. Oh my gosh.
Ooh.
I think we do. Wait a second. Let me try to turn on my. No worries if you can't see it clearly. There's actually a very tiny gray spot here. I'm gonna take a picture of this and put it down in the comment. There is indeed a very, very minor gray spot here. Okay, potentially very minor, but potentially poisonous. Indeed. Okay, so this is my talk for bizarre botany. And tomorrow, oh yeah, since I am here, tomorrow we're gonna need bigger guns. We're gonna need this.
Oh.
I'll leave you to think about what we're gonna talk about tomorrow. Two animals that scares a lot of people, and we're gonna, demystify some of them. Okay, so I'll see you all again tomorrow. Any questions? No questions? Okay.
There are some questions on the comment, but I think
I'll respond to them later. No worries. Let's
as
go.
you've done before.
Okay, like and tomorrow, brace yourself. Yeah, it's gonna be a wild ride.
That's right. So tomorrow is going to be creepy crawlies. That is the theme. And it's up to you to decide what is creepy crawlies all about. Okay. So you kind of have an idea. You see the hook there. Those who know what is going to happen. And I think if you have your kids watching tomorrow, they're gonna really enjoy watching this one. But again, safety first, okay? So we will do things in a safe way. Yes. So yeah, thank you so much everybody for watching. And again, for those who caught in late and you want to catch up, this will be on Facebook, I guess, indefinitely. So you can watch it, spread it, the word out to your friends, and we can learn a lot more. I have learned some wonderful things about plants today, especially the pitcher plant, which I realized I really don't know much about. So thank you so much for today's lecture. And tomorrow we're going to talk all about creepy crawlies and have some fun with animals. So thank you again, and we'll see you guys tomorrow at 7 p.m. Thank you, doctor. We'll see you tomorrow. Bye.